The Yellowstone ecosystem is a really complex ecosystem to management. It's basically a management nightmare. As we've talked about, there are a number of federal agencies which are involved in management of the ecosystem. Plus, you have agencies and governments, city governments, county governments, and so on, of 3 different states involved. So you have national interests as well as local interests, which sometimes are at odds with one another, and you have, politics and economics involved in this as well. But as a scientist, as a biologist or ecologist, really we want the management decisions to be made, not so much on political or economic interests, but really on good, sound science. So it's really important as we learn about the Yellowstone ecosystem to have a good foundation in science and understand what good science is and be able to recognize it. So science then is a way of knowing that utilizes a process to generate a body of knowledge. So again, there's this dual nature, process and the body of knowledge. And what I want to begin talking about first is this process of science which generates that body of knowledge. So this begins in with gathering information or getting experience. So again, with people who work up in that ecosystem, they're going to be exposed to a variety of things. They're going to make observations. This will occasionally lead to questions. And then you talk to colleagues, you may do some research, and it's usually pretty easy to get answer to your questions, whether it's about animal behavior, animal interactions, perhaps it has to do with the vegetation that you observe, things like that. So again, this is normal part of science, is getting background in an area. But typically then you will run into areas where there really are no answers to the question, or at least no one has found an answer to the question that you're posing. And at that point then you will pose an hypothesis. So a hypothesis then is a possible explanation to a question. And ideally, that is disprovable and it needs to be testable if it's going to be a scientific hypothesis. And generally, hypotheses are very specific or narrow in scope. So again, as part of this process then, once you have your hypothesis, we could just choose to accept our hypothesis. Hey, this is rational, this makes sense, I'll just go ahead and assume that is true. But in sciences, and this is what really distinguishes sciences from other way of knowing is that we have to test our idea. We must test our hypothesis. So one of the ways that we test an hypothesis then is in what's known as a controlled study or an experiment. Now, not all hypotheses lend themselves to this, but some do. And when possible, we will use this kind of a test. So there are certain elements to an experiment or controlled study. I'll just briefly describe them, and then we'll look at a couple of examples where we apply these terms. So the independent variable then is that thing that you're testing, what you are manipulating. It's at the crux of whatever your question happens to be. The dependent variable, and again, we want to set this up so that there will be a single independent variable. Whenever possible, we want a single independent variable. Dependent variables are affected by the independent variable, and there may be a number of dependent variables. And most controlled studies can be put into the statement, what is the effect of blank on blank? And the first blank then will be the independent variable. The second blank will be the dependent variable, when you can put it in that kind of a phrase. Then we'll have at least 2 groups, an experimental group or groups that are exposed to the independent variable, and then generally a single control group in which the independent variable is withheld. Now again, not all kinds of experiments will set themselves up for this. For example, if you were looking at the effect of temperature on some behavior or something, you can't withhold temperature. There's always some temperature. It may be hot or cold. So in those kinds of situations, you'll just arbitrarily assign one group to be the control group and one to be the experimental group. But generally, the control group is not exposed to your independent variable. And then last but not least, we want to treat the 2 groups, the experimental and control group, exactly the same except for the one variable, the independent variable. So all of the variables that would be the same in our 2 groups, experimental and control groups, we would want to be consistent. We call those controlled variables. So let's look at an example. Let's say that you've done some research and and this is actually a true effect up in Yellowstone, in that the swan population, the trumpeter swan population, is in decline. And most ecologists attribute that to predation by coyotes, wolves, other small predators. But let's say that you've been doing some research on this decline in swan population, and you happen to notice that there's a correlation between snowmobile use in the park in the winter which has been increasing over the last 30 years, and that correlates with a 30-year decline in the swan population. And so you wonder if there might be a correlation there. And what is known about snowmobiles is that they are basically dirty. They're mostly two-cycle, although the park is making some changes in regulations regarding snowmobiles. But historically, they've been two-cycle engines. They're quite polluting. They produce a lot of pollutants in their exhaust. And these pollutants are hydrocarbons, basically part of the exhaust, actually unburned gasoline, other kinds of components in that exhaust. And so that builds up on the snow. When the snow melts, that runs into the ponds and rivers and streams and pollutes the ecosystem. So anyway, so you're curious if there might be a correlation between hydrocarbons in the park and the swan decline, and you're wondering if hydrocarbons affect development of birds. So rather than use swan eggs, you decided it would be easier and better on the swan population if you tested this in chicken eggs. So you devise an experiment, and let's also say that in devising this experiment, you realize that these hydrocarbons, gasoline and components, don't really dissolve well in water. So you find that you're going to actually have to dissolve these and mix these in a dilute solution of alcohol to make this work very well. So what you end up doing then is getting a group of eggs. Let's say you get 100 eggs, and you punch a small hole in the air sac of these eggs, and you inject a mixture of hydrocarbons with alcohol. And you inject that into the air cell. Those will diffuse into the egg, and then you seal up the hole in the egg. And so let's say you prepare, again, 100 eggs that way. Then incubate them. Well, now you need a control group. That's your experimental group. So again, our independent variable is going to be hydrocarbons. We're interested in the effect of hydrocarbons on our dependent variable, which in this case will be development. So we're interested in the effect of hydrocarbons on development. So we have our experimental group being exposed to the hydrocarbons. We now need a control group. So again, what will that control group look like? So again, we've had our experimental group. We need a control group now. So keep in mind that we want the control group to be treated exactly the same. We only want a single variable. So I think intuitively you might say, well, let's just take 100 eggs and incubate 100 eggs without doing anything to them. But if we had 100 eggs that we didn't manipulate in any way, we would end up with more than one variable. We poked a hole in an egg. Does that affect development? I don't know. I don't want to know that. I want to know if hydrocarbons affect it. We injected alcohol. Does that affect development? I don't know. That's not what I want to test. So you would want to, again, set up a control group that's treated identically, except for not exposing them to hydrocarbons. So you'd want to take a group of eggs here, again, punch a hole in the egg, the air sac. You'd want to inject alcohol without the hydrocarbons this time, because again, does punching a hole affect development? I don't know. Does alcohol affect development? I don't know. That's not what I want to test. And then we'd seal up the hole and incubate these, and we'd want to incubate them under identical conditions if at all possible. So at the end of this kind of a control, then we would have only a single variable, which would be exposure to hydrocarbons. And that way, then, if we have a difference in the control and experimental groups, we can trace the difference back to that single variable. Okay, then everything again that we do the same, so we'd want to use the same breed of chicken, the same incubator, temperature, humidity, all of that would be controlled variables. Now, we can also apply these kind of studies to ecosystem studies, natural studies. It doesn't have to be a laboratory type of study. Again, another situation that was observed in the Yellowstone ecosystem and led to the eventual reintroduction of the wolf is that without wolves, the ungulate population exploded, particularly the elk. Huge elk populations. And again, coincidental to that, what was realized is that the aspen population was changing. Trees were getting old. There were no young aspens. There were really no aspens younger than about 15 years of age. So again, ecologists began to suspect then that this exploding elk population was at the root of the problem. They were eating the aspen. They were observed eating the aspen, and it was thought that they were the primary problem. But there are other ungulates up there as well. So again, we have bison, we have deer, moose also could potentially be the problem. And so again, when the idea was being floated, well, you know what? We need to reintroduce a top-end predator. We need to reintroduce the wolves to control these ungulates. The question was put to them, well, let's see the evidence. Let's see more than just observations. Let's do a controlled study. So again, it was suspected that elk were the primary problem, but they were interested really in all of the ungulates. So again, putting this into the idea of a controlled study, what is the effect of ungulate foraging on aspen growth? gives us our independent and dependent variables. And now we need experimental and control groups. And so basically what they did was set up pens or set up fencing around aspen stands. And so what we had then was one area where there was field fencing that had no gaps in it. And so, when I talk about none here, what I'm telling you is that no ungulates could get in there. Small mammals like chipmunks and so on could get through the field fencing, but none of the large ungulates. And then you had another fencing then that did have gaps in it. So there were, posts that had spaces in it. Deer could move freely in and out of these acreages, but the openings were too narrow for elk. Elk could not get through. And then you had other fencing that had still larger openings that allowed the elk and deer to move through, but too small for bison. And then you just had areas basically that were unpenned, and really all of the ungulates could get in there. So this was our control group. And, this was our experimental patch, as was this patch, because this is where the elk were. And these really served as our two controls up here, places where we were not going to see the elk. So as it turned out, again, this literally has run for decades, and what we have found is that where you don't have elk, you do have young aspens. So anyway, this then gave us the evidence to say, this elk population is a problem. We need to limit it. We need to bring in a predator that's going to feed on elk. And that's what justified bringing in the wolf population. So again, the control variables here would be basically you want to make sure that they're in the same location. Basically, you're looking at the same aspen stand, and then you're dividing that up into different fenced regions so that the climate effects, temperature, humidity, rainfall would all be the same. Sometimes the control study doesn't really work. Sometimes you just need to make more observations. One of the hypotheses that had been, and theories really, that for the ecosystem was that when you look at transitions in ecosystems, it was thought that aspens always came first, and then eventually transitioned into evergreen forests, lodgepole pines. But and so again, that would be your hypothesis that after the 1988 fires, which decimated the park, we were expecting to see a lot of aspens first and then conifers later. But what we ended up observing was that where there had been lodgepole pines grew back, and where there had been aspens, aspens grew back. So this is one situation where the observations didn't match what our hypothesis was. But you really wouldn't call that a controlled study per se. All right, so again, kind of the next step then is once you've done the test, you want to statistically or objectively evaluate your data. And one reality is that when you look at data sets, is that data sets will always have differences. For example, if I looked at two different classes, if I took the average height of one class and the average height of another class, there's going to be a difference there. But one of the issues that we have to determine is the difference in a data set, two different data sets rather, is that just due to chance? For example, if I look at different heights in different classes, is there a cause for that difference? Or is that just what we call a sampling artifact? Is that just randomness? Is that just the way it worked out that time? So just because there's a difference, it doesn't necessarily mean there was a cause for the difference. And this is one of the dilemmas that we have in the sciences. So again, are the observed differences due to chance, or are they due to some cause? And again, in a controlled study, that would be our independent variable. So we generally can look at this by applying our idea to two general hypotheses. The null hypothesis says or states that any difference in our experimental and control group is due to chance, is due to sampling artifact. It just happened that way. The alternate hypothesis states that the difference in our data sets is due to a cause, due to our independent variable. So again, how can we address this? What do we do? So let's go back to our egg experiment. So let's say that in our experimental group, that only 85 of the 100 eggs developed normally. And let's say in our control group that was exposed only to the alcohol and not the hydrocarbons, let's say that 92 of those developed normally. So again, there's a difference there. 7 more died when exposed to hydrocarbons. And so you might say, well, then hydrocarbons cause more birth disorders or developmental disorders. But again, in the sciences, we now have to kind of consider, well, I wonder if that difference is just due to randomness, or was it really due to the hydrocarbons? So one of the ways that we would address this is to repeat the experiment over and over again, and there are statistical methods to do that. And then also one of the ways that we can address this question is what if, instead of keeping our data sets separate, what if after we incubated the eggs, somebody had just mixed them up, just randomly mixed them up into two groups of 100, so that I didn't know which ones had come from the experimental group and which had come from the control group. And if I had a certain number of X that developed normally in group 1 and a certain number that developed normally in group 2, which would be Y, if I subtracted those, could I get the same kind of result that I got with my experimental and control groups? So we refer to these as randomization studies. So again, there are statistical techniques to randomize our data or to basically repeat our experiments a number of times. And ultimately, what we have to establish is what we consider a 95% confidence. So scientists don't normally say that we prove it or don't prove it. We generally talk in terms of confidence. And this 95% confidence basically is a way to statistically redo our experiments or manipulate our experiments in a way that if we did them again 100 times, if we did our experiment 100 times, if we got the same kind of results 95 or more times, actually, excuse me, more than 95 times out of 100, so we would have to get the same kind of results 95 times or more per 100, then we would have a 95% confidence that there was a cause for the difference. And if we have at least this 95% confidence that there is a cause for the difference, then we say there's a significant difference in the two groups, and we accept that there is a cause for the difference. So we have enough confidence to say, yes, there was a cause for the difference. Now, once we've done that evaluation, we come back to our hypotheses. We can basically do our evaluation. We can accept our hypothesis, reject our hypothesis, or possibly modify our hypothesis and then do another test. So science is kind of a dynamic thing, but you make your conclusions based on your data. That is crucial. And then, once you've done the work, you want to communicate your results. You don't want to work in a vacuum. And the way that we do this professionally is that we will publish in peer-reviewed journals. We attend scientific conferences, present there. And of course, we're constantly communicating informally with one another, just as anybody would. But as I'll come back to later, really it's the collective intellect which helps us figure out if we've set up a good study. Was this a problem in terms of our design? Are our conclusions skewed? We need to communicate and come to some collective intellectual conclusion. Now, as we look at the products of science then, certainly the data that we generate in our controlled studies and other types of studies would be one of the products. But also the ideas, are the products as well. So hypotheses would be one product of science. And so when we generate hypotheses, the whole point of a hypothesis is that we're trying to test some idea. So initially, hypotheses will be unverified. But if we do it over and over and over again, and we get the same results over and over and over again, then our hypothesis is no longer an unverified hypothesis. We call this a verified hypothesis. And verified hypotheses are factual. We accept them as fact once we've repeated them over and over and over again. So again, I think most people have the idea that a hypothesis is an educated guess, but not really. They start out that way, but over time, we gain confidence in them and they become factual. Theories are built from related hypotheses. And again, it's the same kind of idea. Initially, a theory will be unverified. But when it is repeatedly and accurately tested and predicts accurately over and over and over again, then it again is no longer an unverified theory. It will become a verified theory. And again, in the biological sciences, ecological sciences, we look at verified theories as factual. Again, not just some pie in the sky kind of idea. It may start out that way, but repeated testing gives us confidence. So as I mentioned then, theory is built from related hypotheses, and we'll use something called inductive logic or reasoning to do that. They're usually mechanistic. Theories also usually address the mechanism behind the phenomena you're observing. And then also they're predictive. So you use theory to predict more hypotheses, really, using deductive reasoning. And then, as I mentioned, verified theory is factual. Now, there's some other terms that were used in some of the sciences, principles and laws. We don't really use these terms much in the sciences, but basically they are just statements of observed phenomena. For example, the law of conservation of matter states that matter is neither created nor destroyed in chemical reactions. Doesn't really tell us why, but that's been tested so many times that we accept that as fact. So a principle or law is not necessarily more factual than a theory or a hypothesis. It's just something that's been stated so many times, that it is accepted as fact. And again, in the biological sciences, we don't really use the terms principle or law very much. We just work with theories, verified and unverified theories. So again, as we build hypotheses and theories then, inductive reasoning takes specific ideas and builds to a broad conclusion. And this is how we build theories. We take related hypotheses and build theory with this. So, for example, historically, a very common theory is the cell theory. So when people were looking at organisms, they didn't know what they were composed of. And so if you go back into the 1700s, the early microscopists produced microscopes, began looking at organisms, and they found these structures called cells. And over time, everything that was observed to be composed of cells or cell products. We also eventually observed where cells came from. Cells came from previously existing cells through cell division. So eventually this led to the cell theory, which states just that organisms are composed of cells, cell products, and cells come from previously existing cells. So now you have, through inductive reasoning, this broad conclusion. Organisms are composed of cells, cell products, and cells come from cells. Now you can take that theory and make predictions. So we use deductive reasoning or logic for that. So in deductive reasoning, you'll take some broad concept and make specific predictions. So this can also typically be worded in an if-then kind of statement, although you have to be a little careful in how you do that. So again, using the cell theory, I've never actually looked at this pine tree leaf behind me, that particular tree, but if organisms are composed of cells, then I would predict that that is composed of cells. And I could test that hypothesis very easily by sectioning it and looking at it with a microscope. And I can guarantee you that I would find it is composed of cells. Now, looking at science then, again, and kind of with a broader brush then, what makes good science? When are you observing good science? What is bad science? First of all, science is mechanistic. Okay, when we're doing science, we have to be able to ask ourselves and others the questions, what is the mechanism behind what I observe? If you're not willing to ask that question, if you can't ask that question in your way of knowing, then you're not doing science. So that's where it begins. Secondly, we have to work in the natural world. We have to do work that others can also do. Our experiments have to be duplicated by others. We have to be able to generate data that can be statistically evaluated by others. So I may have a dream about something. I may have a dream that hydrocarbons cause swan mortality, but that's not good science, even if it's a really good dream. I've got to do the test, I have to generate data, and others have to get the same kind of results that I get and reach the same conclusions for that to be good science. So we are stuck in the natural world. So it's magic, God did it. Those are not scientific concepts because those are inherently untestable. So we have to work in the natural world to be able to test our ideas, which is another characteristic of science. And this is what, again, and I think is really the crux of what distinguishes sciences from other types of knowing. You can believe anything you want to believe, but in the sciences, if we have an idea, we have to test it, we have to generate the data, others have to be able to duplicate what we do, they have to reach the same kind of conclusions, and now we may have an idea that's worthy of accepting, quite frankly. But again, in everyday life, we don't have to do that kind of thing. We can believe whatever the heck we want to believe for whatever reason we want to believe it. So again, kind of following the same logic or idea then, science is also evidentiary. So again, I can't have a dream. I have to generate data or something that is manipulable, that people can observe. That's part of the evidence of science. We just can't accept things a priori. And again, I think this goes along with the idea of people, want to be open to ideas. Well, scientists are curious. We love new ideas. You just have to bring the evidence. You have to bring the material. We have to be able to test it too. We have to get the same conclusions. Now we're talking the same language. Now we've got something that we can accept. Another hallmark of science is objectivity. We can't really go in with prejudice. We can't be prejudiced when we evaluate our data. And that's why you want to use mathematical statistical analysis whenever possible. That takes out subjectivity. So again, that is really crucial to good science, is being objective, being open-minded, and let the data tell you what the conclusion is. As I've mentioned a few times with this, then science has to be repeatable. If I do it, if you do the same thing, you should get the same results under the same conditions. If you don't, we've got a problem. Maybe my idea wasn't such a good idea. So again, just really crucial to what good science is. And then ultimately, there are no absolute truths. Our ideas are only as good as the next test. And so again, we're really open to that. We want challenges to ideas. That's what makes a great scientist. The really great scientists have tremendous imaginations, tremendous confidence, and are willing to look at new ideas. But the problem is a lot of times people have ideas, say this is the way it is, but they can't bring any evidence for that. So it's the collective intellect then that really works. You have to be able to communicate your ideas, let others mull your idea, others do what you do, see if they reach the same conclusions. So when we look at the public's skepticism of science, I think there are a lot of reasons for it. One certainly is just a lack of understanding of what science is. Science is a very specific type of knowing. You test your ideas. Also, I think that a lot of people just have the idea that in life, if you make a reasoned conclusion, that they're doing science. And that's not always the case. You may have a very reasoned, sound conclusion. It may be completely wrong. So again, just because something's logical doesn't mean it's scientific. What makes it scientific is that it's been put to a test, it's stood up to the test, and been accepted, and the collective intellect has been able to test it and verify it as well. Also, I think the general public confuses being open with acceptance. So in other words, in the sciences, we don't accept an idea until it has been tested, as I've mentioned a million times already. So it would be nice to just believe in something because we want to believe it, but that's not the way it works. And then ultimately, it's the clash of religion and science, which is ultimately the biggest problem, because really, religion is all about faith. It's the lack of objectivity. It's the lack of testing, quite honestly. And science is just the opposite of that. We can't have faith in our results. We must be open-minded. So unfortunately, there is this kind of clash between the two disciplines. And I'll hopefully talk more about that in a future module.